This nanocluster, I'll come back to this slide, but let me quickly go to this and then I'll come back to metal nanocluster. All of you are aware of experiments, right? Chemistry is fun, because we do the experiments, we go to the lab, deal with test tubes, chemicals, and mix them, do magical stuff, starting from changing colors to creating some crystals to seeing something happening there, bubbling, bubbles come out of a test tube or a beaker. So that's fun, right? But then, if you have the superpower of seeing what happens inside the test tube, right? In principle, you can have a microscope, very powerful microscope, and you zoom in and see at the molecular level and see what happens, or you do something called molecular simulation, right? So in situ experiments means you see things live, things are happening, and then you record it by using a powerful microscopy, and then you see what is, how the molecules move around, and the chemistry, the reactions, surface reconstruction, all these things you'll be able to see it by doing experiments. But then if you want to zoom in further and see the atoms and molecules, how do they move around, react with each other within, let's say, time scale of nanoseconds or milliseconds, right? So then many of these experiments are kind of limited because the sensitivity of those instruments are not to that level where you see nanosecond time scale things, or even, let's say, the atomistic details. I mean, there are sophisticated instrument for sure by which you can do it, but for now, let's say, a very convenient way is to do the modeling, or it's called simulation, computer simulations. The word simulation means that you mimic a real process, meaning that the way we are sitting here, if I ask you, okay, just stand up and roam around, that's a process, right? So initially we are sitting in an ordered phase. So assume this is a crystalline phase, and if I ask you to do, stand up and roam around, that could be possibly molten state, right? So we study from ordered to disordered state, and this can be modeled in principle on the computer. So each of you would be a particle or an atom, and then we interact with each other. I mean, we don't collide. There's a less chance that we collide or we sit on top of each other. We don't do that. So eventually what you see that there are some interactions, or let's say the kind of our comfort zone. So the way we are sitting is like we are maintaining some distance. Okay, we are not sitting on top of each other. That's not allowed because that's a high energy process. And then, so that's kind of interaction field that we think of. And then when we disperse and then move around, then there is another way of interactions, right, at two different conditions. So this can be in principle mimicked or modeled using computer, okay, and then you see the same process depending on some conditions. So computer simulations is you utilize computer to simulate a real process. That's the background, okay. Now a glass of water, a bottle of water here, okay. What do we see here? So in this water, what do we see? Can you see anything here inside? Now I give you a superpower. What would you see here? Assume that you have got the superpower, sci-fi movie. What did you see here? Hydrogen and oxygen, right? And that superpower is textbook, essentially, right? We know the textbook, and from there we know that there are oxygen, hydrogen, they're bonded with each other, all these geometrical parameters, they form hydrogen bond, and it's in liquid state at room temperature and pressure. Okay? But then that superpower in principle is textbook and then imagination. Let's see that textbook, certain set of equations and a bit of programming would probably help you to see what is happening inside this water. So what we do, what we do here is we zoom in some portion of it from the glass of water. We run molecular dynamic simulations. Molecular is again molecules. Dynamics means the molecules are moving around. It's a dynamic process. Simulations, I told you, it's a mimicking the real process. And then there are a set of equations and algorithms. You write the code on computer, and then what you see is something magical, right? So it's dynamic. Okay. Now look at this. Water molecules are moving around, and then they are interacting with each other. At some point they form this hydrogen bonds. You probably see this broken kind of transient bonds, right? They're forming hydrogen bond, and that's how it's liquid at room temperature pressure. So the way you could imagine here, or probably you would utilize a microscope, you do the same thing on computer to get the stuff like this. So what is next? Do you believe this? From textbook, yes, we believe that okay, water molecules, they interact through hydrogen bonds, but then how do you quantify, how do you validate it, that this is water? What you do is, after running the simulation, you analyze the data. So you analyze the trajectory and calculate certain properties like density. What would be the density of water at room temperature and pressure? And then you see whether this density with what we calculate from our molecular dynamics is matching with experiment. If it matches to certain extent with some, let's say, fourth decimal point, then we believe okay, whatever we have done on computer is correct. So what is fun in this? I mean, we know the density of water from textbook. Why do we need molecular dynamics simulation? What if I tell you that I want to simulate water in some interstellar condition? Water is one of the mysterious chemical in the universe. Probably you guys know, right? We didn't solve water yet. We know only a little bit about it. Water can give you nightmares if you think in different kind of situations. So if you want to study water in interstellar conditions or at high temperature, high pressure, then you don't do it every day. You don't go to the high pressure setup and study water. What you do is you do modeling. So you study it by doing computer simulations, understand its behavior properties, and then you make a list of things. Okay, I do this experiment, this looks interesting, and you do that experiment. So that saves your time, money, as well as effort. Okay, so the computer modeling complements it does a prediction as well as it complements your planning for experiments, as well as when you do experiment, you come back and do the modeling, and this is a kind of back and forth transitions. Okay. Now, how do we model water? That movie is very fantastic, right? I mean, we saw the molecules moving around. What is the underlying physics behind it? So now probably this is very, again, going back to textbook, 6th, 7th standard, we have studied mechanics, right? When we walk on the street, right? If I'm standing here, you probably guess what would be my next move. Okay, it's either right, one step, or I can go left. There's less likely that I can fly, right? Less likely that I can go up or I go down. So what decides my movement? Or my initial position is here, what decides that I move this side or this side? Can you guess? Space is, of course, I mean, this side is also space available, but you see, I'm not going that side. It's a force, right? It's a force acting on me. Let's say, assume that there is some force on top of my head which decides whether I go this side or that side. You'll probably feel the force when the lecture is over, we go for tea. Your force will allow you to move only this direction. Nobody would go, this direction, go for tea. Okay, you have to be quantum particle to tunnel and then appear there. So this is whole classical mechanics, Newtonian mechanics. So if you know the position at time 0, you can predict your position at time t, right? And then you have studied V equal to U plus Ft, all these things, right? S equal to blah blah blah. But then now that you are grown up kids, you make your life complicated. It's true, right? And those are simple equations, but you see here, it looks little complicated. With r double dot is the acceleration, is the force acting on each particle. This is mass of a particle, and then again you write the force on each particle as a negative gradient of the potential. What do you mean by potential? Is the way we interact. So the way we are sitting here, we have some interaction. So if you are sitting next to your friend, then most likely you have attractive interaction, you like it. If you are sitting beside your enemy or you don't like the friend, then you repel each other, right? And then you combine this all this kind of things, then you write the potential of this classroom. And then you take the derivative of that potential with respect to each one of you's position, then you get the force. So that means you can calculate the force right now, the force acting on you, that decides your movement like this. So a person moving, it's very simple. What about this? everybody is having some force. Whenever you go to crowded place, then you see nobody would be standing there. Most likely they are moving in certain directions. So force is a direction, okay, it's a vector. So all these guys are moving in certain direction. It's not like really chaotic. There is some flow, non-equilibrium process. So it's called crowd, the crowd that we know. Now you go to molecular level, then you can call this one as a molecular crowd. It's the same mechanics is being followed. Okay. Now this one, our interactions, we can kind of comprehend the kind of way the way we interact with each other. What about these molecules? How do they interact with each other? Assume that I have four atoms. Okay, it's just like this. How would they interact? Any guess? Four atoms, no charge. Leonard Jones or Van der Waals, you have heard of, right? Leonard Jones and Van der Waals, they'll have Van der Waals interaction. And if I add some charge on top of it, just labeling it, somebody has plus some charge, someone has minus, then another interaction is electrostatic Coulombic. So then we can write the equation. So this is our Leonard Jones and this is our Coulombic interaction, right? Now let's make this complicated. Draw a molecule. So from these atoms, what I did is I added them by bonds. Now it's a molecule having four atoms. It's an imaginary molecule, okay? Butane, let's say butane without hydrogens. Then what kind of things you can think of? Now the atoms are not free to move. You have added another constraint, and because of that it will feel some potential energy and it will feel some forces will be different. Then you can write something like spring. So assume that two atoms are connected by spring, then you can do a simple harmonic motion kind of thing and you can write this equation. So this, if you think of the Hooke's law, right, force, and then you take the, what do you do, integration, you get half kx square. This is harmonic like spring. Similarly, you think about this angle, they also go like this kind of motion, angular. And then you have the dihedral, because you have a four atom. So dihedral means that you put two atoms on plane, three atoms on a plane, and the three another plane, and you take the norm, and the angle between the two norms is a dihedral. Okay. So then now we have this non-bonded interaction. That means there were no bonds, and we have bonded, or we call it intramolecular, within a molecule. Okay. You combine them, you get your total potential. So this is potential energy, okay, which is non-bonded, these two, and bonded is a total potential energy. If you get the total potential energy, now can we calculate the forces? Answer is yes, we know it. This is the total potential energy. You take the derivative with respect to each position of atom, you get the forces, and then you apply this force here in the Newton's equation of motion to update your positions and the velocities. That's it. So my position is here. Assume some initial velocity at delta t time. I move here, I changed my position, and now I change my velocity also. Okay, and it is being guided by this force that we get from the potential energy interactions. Okay, so all the way we interact with each other decides where we are. So they say that, I always use this thing in the class, that find your friends, you will be in equilibrium. If you are with your enemies, you don't like the people, then you'll be in non-equilibrium. Okay, so the force will guide you, whether you'll be doing some extraordinary thing, that is decided by your environment, the people whom you interact with. That's very important. Now this, these terms like the spring constant, equilibrium bond distance, equilibrium angle, for water we know the equilibrium OH bond distance 0.97 angstrom, or the angle 105 degree, right? Then associated with this some spring constant. So that allows how flexible this one, how flexible this angle is. These are called force field parameters. So these are parameters that you obtain from textbook, and again these are derived from either XRD, OH bond length, you get it from X-ray diffraction. You do IR, infrared spectroscopy, you get this kind of spring constant, stretching, bending, those modes you can calculate. So these are called force field parameters. So these parameters you need to supply to the computer. When you say that I want to simulate a water molecule, then you supply the equilibrium bond length, the spring constant, equilibrium angle, and then some sigma epsilon values for the Lennard zones and the charges, atomic charges. Then what you do is, typically this is a bit advanced stuff, that you use something called integrators using finite difference method to update positions and velocities. And those of you who would study StatMech probably encounter this algorithms. That's all about molecular dynamics. You write the potential energy function, take the derivatives, use integrators to update position and momentum. Now, how many of you are aware of very old movies, black and white, how they used to shoot the movies? Any idea? Satyajit Ray, that era. Okay. Charlie Chaplin, even that era. I mean, I'm just a little, forward. They would take some snapshot with a camera. They would take some pictures, still pictures, and they would create or combine them into reels, right? Film. And somebody would take this film to go to theater, and somebody would rotate it and project it on the screen. Have you seen this kind of thing with the sound? It would sound. Molecular dynamics is something like this. At time 0, right now I take a picture of this classroom. After some time I take another picture. Okay, right now it is not changing much, but if I ask you to roam around, I take snapshot every time. So time t equal to 0, 1 femtosecond, 2 femtosecond, 3 femtosecond. I take thousands of this snapshot, I combine them and play it. That's what we saw in the water molecule moving around. Each of this frame is called a configuration. Okay, it's called configuration, and then you update it. Okay. Now, what do we do in molecular dynamics? We have a system, this classroom. Okay. We need the coordinates. So each one of you has a coordinate. Do you feel it? What is your reference? You look at any corner, that is XYZ Cartesian space. Look at any corner. Maybe this is not the ideal one. This one, you take it XYZ. With respect to the origin 0, 0, 0, assume you can think of a vector from 0, 0, 0 to your location. That's your position R. Okay, so this classroom, each one of us has one position associated with us. What about molecules? Water molecules, what would be the coordinates? We can write them. You put water, oxygen at the origin, and then you orient accordingly, and then you can write it exactly, because you know the bond distance and the the angle also. For proteins, let's say, proteins, right? Amino acids, long chain. Where do you get the coordinates? You go to protein data bank. So the structures are solved using XRD or NMR. Download it, then you will see all the atoms and corresponding XYZ coordinates. Or if you are interested in modeling, let's say, solid state systems like crystal, sodium chloride, okay, rock salt, you want to simulate, you need to go to CCDC database and then get the CIF file. It also has coordinates. Or you can write your own script, scripts to get the coordinates, or other sources. You can use ChatGPT. Ask ChatGPT, okay, give me coordinates of 333 supercell of sodium chloride. It will give you the coordinates. That would be your initial set of positions. Right now, the way we are sitting, then what you do is you define simulation parameters, number of particles. So if we are now, let's say 50 of us, that's the number of particle. Initial temperature and pressure, room temperature and pressure. Let's say we want to simulate this classroom. Integration time step, that means what would be your frame. Okay, if I want to wait for 1 second, so t equal to 0, 1 second, 2 second, 3 second, 4 second, this is my time step. That is 1 second I am taking a snapshot. That's called integration time step. Then we assign some velocities. If I suddenly ask you to roam around, what would be your velocity? Can you guess? Anyone? And what is the textbook chapter that you study to get the velocities? Huh? Tell me. 2 kilometers per hour. Okay, in this, somebody cannot go 100 kilometers per hour. Why? Because of collision frequency, and then you go to a chapter kinetic theory of gas, and that says typically you follow this Maxwell-Boltzmann distribution. That corresponds to temperature in this room. Okay, if all of us can keep on running 50 kilometers per hour, imaginary, the room temperature would go high because kinetic energy is increasing, and you think 3 by 2 kBT and typically mv square, half mv square. That's the combination between these two. Okay, so we need to respect this one to get the temperature right, and then what we do is we assign the velocities based on Maxwell-Boltzmann. Then we calculate interacting forces. By now you know how we calculate the forces. Then we run, molecular dynamics, where you know the initial position, and then after delta t, which is 1 second, how your position would be, and your velocities also you update accordingly. Once you do this, you get a trajectory, that means 1000 frames, you analyze it and see the properties of it. Okay, so this is basic of the molecular dynamics. Now, so far we discussed about us or atoms or molecules. Did we talk about electrons? No, we didn't care. We thought that okay, we model atoms connected by springs. So that's a classical description. That means we don't know what is happening inside one atom. If you want to zoom in and see what is happening inside atoms, then you'll immediately encounter electrons. Electrons are important because if you want to study normal water, that is okay. But if you want to study a chemical reaction happening inside this water, okay, then you have to deal with electrons. So how do we deal with electrons? You know density functional theory, some of you have heard of. Okay, so what we do here is to incorporate electrons into our calculation. We incorporate how the electrons interact with each other, or how the electrons interact with nucleus. So nucleus electron and electron interactions we have to incorporate, and there is a convenient way of doing it, is called density functional theory for a many electron many body problem. Okay, if you have no electrons, we have already discussed, this is a classical description, and with electrons we have this quantum mechanical or quantum chemical description. And there is a method called AIMD, ab initio molecular dynamics, which combines this classical motion along with your electronic motion. That means, assume assume that you guys are like quantum particle. Many things would happen, right, which we even cannot imagine, or we can imagine but we cannot comprehend. If you are quantum particle, we coexist here as well as in your hostel. Or once the lecture is over, you can tunnel from here and appear there in the T region. Okay, it's like a ghost. That's a wave particle, right? Wave particle duality that you talk about. So we combine both classical as well as quantum to study chemical reaction. That means your electronic states are also changing when you propagate your molecular dynamics simulation. That means you think about SN2 reaction, you have the reactant, okay, there's a attacking group and there's a leaving group. So that this is one electronic state, and when the product forms, then the reactant, that means the incoming ligand, attacked, and the outgoing group got removed from there. So that's a different state. Reactant and product connected by one transition state. Okay, so this is what we model in computer. When we need to deal with chemical reaction, we need to incorporate electrons. Now, if we deal with electrons, assume that hydrogen atom is okay, we can write it, we can solve, right? The Schrodinger equation. Next, helium or H2 plus, textbook, we can do it fine. Helium, extremely difficult, right? You can write it, maybe months or something you derive, but then you end up with something electron-electron correlation. That's where the problem is. Now think about carbon, CnHs. Now you go to palladium, lead. You see where we are making the kind of thing, life complicated. And if you want to calculate all the electrons and their interactions, it's super complex as well as time consuming. Even if you use computers of, let's say, probably you spend $1 billion and you purchase one computer, that too also would struggle with solving some of the problems that you need to run simulations of huge, let's say, system having heavy metals. That's difficult. Now, you remember I was talking about a time step or time scale. Typically, we in the atomic scale or atomic or molecular level, we deal with femtosecond time scale, picosecond, okay, or nanosecond. To run a simulation of 10 picosecond, including electrons, typically we need a week time with if you use GPUs. But 10 picosecond, what happens? Do you think anything happens in 10 picosecond except some rotation and something like that? No. Reactions, the experimental lab, you put some sodium chloride, supersaturated solution, stir it, go for a coffee and come back, then you will see the crystal. What is the time typically? Half an hour, right? Or if you have a very quick, the process, that means the reaction would happen in seconds. So there are experiments where it's called a chemical clock, right? It changes the color, and that changes in seconds. But there are reactions that even take place in millisecond time scale or even microsecond. But you see the gap. I'm talking about picosecond, and the reactions are taking place in microsecond or millisecond. We cannot do it. If you want to run a microsecond time scale simulation using quantum chemistry, the electronic description, this life is not enough. Okay, meaning that you have to take both many times. You have to be born and then keep on running the simulation. So that would be the time scale. There is a way out. Let's not run the simulations with all electrons every time. Take the help of a neural network. Okay, so neural network means that I know the energy of this classroom at a given condition, number of particles, volume and temperature, which is called canonical ensemble. If you study StatMath, we know the energy of the entire classroom. We divide the energy by each person. So each one of us has some energy. Okay, that energy could be classical energy or it could be quantum included. We ask the neural network to remember this set of particles at this state, this is the energy. We change the configuration, again we supply the energy. So that means now we are teaching somebody that, okay, for this configuration this is the energy, store it with you. At some point I'll ask you, what is the energy of this configuration which I give you now? Then that person would probably tell you, okay, this is energy. So that means what we are doing is essentially training, okay, training somebody. So assume that we are training a kid, the horses and the goats and those kind of things, right? So depending on the look, the some features that kid is learning, these are the horses and these are the goats probably. At some point we take that kid to a field, mix horses and goats, that kid would be able to distinguish between those two animals. It's the same thing. And 2014 Nobel Prize was given to physicist and chemist. So to these guys, Hopfield and Hinton, for designing this kind of architecture. So we have the neurons in our body, in our brain. These neurons can be written in terms of a mathematical, you know, uh, architecture, which is called perceptron. So you have a dendrons that accept your inputs, and then axons, it passes the information to the next neuron. We have the same mechanism. We have some inputs, goes to the neural network, it processes something and gives it to the next neuron, and you combine this, you get a neural network. This is called neural network. So that means you have input layer which takes the inputs. So that's a handwritten number. It does something mathematics and it gives you an output which is a number. And we are living in this era, right? AI. From starting from waking up in the morning to going to bed, you cannot skip AI. Go back and think about it. You cannot escape from AI. Anyway, so let's see how we apply this stuff. So what are our interests? So we apply machine learning for molecular property prediction. We can predict energies, we can predict spectra, reactivity, machine learned potentials for molecular dynamics and material simulations. So now in simulations we need to calculate the energy every time. What if we tell the network to store the energies and remember, and we don't calculate the energies? That means the molecular dynamics would be faster because we are not calculating every time. So that way we can apply machine learning to molecular dynamics. We can utilize those for drug discovery. AlphaFold, all of you have heard of AlphaFold. It kind of solved 50 years old, the problem of understanding protein folding. So that means you start with an amino acid sequence, you give the sequences, you run the AlphaFold code, it would give you a protein folded structure. Okay, we can utilize those kind of architecture to do drug design. The COVID medicine, it came within few months. It's only possible because of this machine learning models, which would otherwise take 10 years. Okay, that's the impact of AI. We can think of retrosynthesis planning. What we do essentially in neural network, we are training the network with some information. We have studied name reaction, 30s, 40s, 60 name reaction we remember, and then we go to exam and then we see this and we apply, okay, that reaction, name reaction would apply here. Now machines can do that. Train the machine with all the name reactions. Now it can give you a retrosynthetic pathway, go back and this would be your reactants, and go back and forth, and even it can predict whether this molecule can be synthesized in the lab or not. That's the power of ML. Material design, okay, you want to design something more, let's say that can sustain some what do you call it, pressure, high temperature, high pressure. For many applications you can design it based on some existing data. Self-driving labs, whatever we do in experiments, okay, many times what we do, I mean, I'm not so familiar with experiments, so pardon me if I say something not so pleasant for experimentalists, but let's say imagine that you have a set of reactions that you plan to do tonight or this afternoon, hundreds of reactions, and that's a kind of, you want to do the combinations A plus B plus C with certain proportion, some, let's say, mole fraction. That part can be in principle done by a robot, and in that same time you spend something to do something more creative, more complex. Isn't it a good idea? Then instead of spending 2 hours to do all the reactions, let the robot do it. So now the self-driving labs are kind of, people are building the self-driving labs. So they are just setting up one code on the computer. That code gives the commands to the robot, and robot is doing all the chemistry experiment. In that time you are thinking about something very different, very creative than doing that labor work, typical work. Okay. Yes.
Driving labs, like maybe it is possible that we will not be able to get some anomalic results. Like sometimes we get something that we don't expect, so that will never come in self.
Yeah, it will never come. So self-driving lab is something to do the, to replace this laborious job that you do typically repetitive. But interpretation and all, that's where we need our intervention. To discover something, you need human. To do something, you can replace it by robot. That's the difference. Okay. Now, in molecular dynamics context, what we do is, let's say this is kind of a neural network. What we do here is we incorporate the coordinates, because we needed the coordinates. You remember when we run molecular dynamics, energies and forces we supply to the neural network so that it learns and gives me the energy. So this Beller and Parinello in 2007, they thought that potential energy surface can be learned using a neural network, and that's how we kind of bypass calculating all the energies and forces, in the simulation. That way we can accelerate the process. Okay, slowly I am going to machine learning driven or AI driven molecular dynamics setup, and this allows us to do simulations almost at the accuracy with ab initio quality, that means DFT level accurate simulations, but the speed or efficiency is almost comparable to classical simulation. That's the power of this neural network. And we utilize this kind of codes which already exist, openly available, to run the simulations. Along with this, we write our own codes to do a lot of other stuff. Okay. So now, if you want to do something fully quantum chemical or quantum mechanical, you take the help of DFT. DFT, all of you have heard of. You do Gaussian, okay. But Gaussian calculations or DFT calculations are restricted to few tens of atoms, 50 atoms, 60 atoms, 100, still okay. Beyond 100, you need to run it for 1 week, 200, maybe 3 months, then you go to 1000, several years. Okay, that would be your scaling. With machine learning potential, you can accelerate the process. The one that would probably take 3 months, now you can do things in 1 week with the machine learning. But the macroscopic observation, again I told you, this takes place in microsecond on millisecond time scale. There what we need to do is we need to accelerate the simulation. So typical molecular dynamics simulations are run up to microsecond or millisecond with super powerful computers. Okay. But then if the process takes place in 1 second, then you cannot study it. So for that we need to again add something called enhanced sampling. The word enhanced means you are enhancing something in the system. You are enhancing fluctuations. Meaning that we are sitting like this in a crystal phase. I am the person, this enhanced sampling guy. I am giving you an instruction that okay, now stand up and roam around. What did I do? I just added something called bias. I added to verbal communication, I changed your forces by this instruction. This is called enhanced sampling. That means now the way you are sitting here, you can write your potential energy the way we did with equations. You can get your kinetic energy based on your equipartition principle. Then you write your total Hamiltonian. Total Hamiltonian is kinetic energy plus potential energy. Now this is not enough. We are adding some bias into the system externally, and that does the job. This is called machine learning molecular dynamics plus enhanced sampling. I'll discuss what do you mean by bias. Okay. Now for a while we are changing this tone a little bit. So far we talked about atoms and molecules. But then if somebody says that I want to study nanobio system, very big system, okay, then you cannot even think of simulating it atomistic level. So you need to go to something called coarse grain level. So that means if you think of this benzene here, now it has 6 atoms. If you forget about hydrogens, you can run a simulation with a 6 atom benzene. But if you have 1 million, 1 billion benzene, then what you do is instead of taking 6 atoms, you take only 3 atoms. So you combine these 2 carbon by 1 bead. It's called coarse grain models. So that means out of 50, we make some cluster. These 2 guys would have 1, these 2 will have 1. So that means we are reducing the number of degrees of freedom in the system or number of particles. That is another way of accelerating the process. Okay. So 2 ways. One is you add the bias. Another way is you reduce the number of particles in the system by doing this coarse graining. And this is a standard Martini. We have recently developed a method, again using neural network, machine learning method, to go from atomistic description, this is azobenzene, to this kind of model where you see the number has significantly reduced without compromising any property. Okay. Now this is kind of a summary for now. We have a potential model which is accurate and fast, neural network potential. We have coarse grain models, the one that we just saw, to simulate large macromolecules. But what about time scale? How long do we need to simulate to study? If we want to study self assembly, going to nanocluster, how nanoparticles come together and create a large macromolecular complex. Can we study it? Chemical transformation, catalysis, if you want to do. Okay, heterogeneous catalysis. You have heard of this hydrogenation reaction on platinum, right? So if we want to model, how do we do that? Drug delivery. This is also another word, right? You have This, you inject something, and there's a drug carrier that carries the molecule to the, let's say, cancerous cells. How do we model it using computer? So these processes take place in at longer time scale. Now coming to enhanced sampling. If you want to simulate a process or a system in equilibrium, that means the water. So that means you have one basin. So I took one example. So that means you stay here all the time. This is simple water. But if you want to simulate a process, let's say chemical reaction, which involve this kind of potential energy surface, multiple steps, transitions. So you are in this basin, reactant. The other basin is on the other side of the mountain, and there are several paths. You have studied this, right? Transition state theory, chemical reaction, SN2, SN1, those kind of. It can have intermediates. It may not have intermediate. Only transition state that goes from one end to the other end. How do you deal with this kind of multi-state problem? Or a typical textbook protein folding scenario. Let's say this is your folded state. You come out of it, you go to unfolded or misfolded state. Or this is a typical textbook. This is a folded basin, and outside of it is unfolded region. How do we simulate this kind of process? And there we apply typical, the one that I told you. This is your normal Hamiltonian, and then you add the bias. This is called enhanced sampling method. Okay, molecular dynamics, AIMD, ab initio molecular dynamics, machine learning, and now I have enhanced sampling. These are 4 components. So if you are here, let's say, imagine your SN2 reaction, you are here, you remain stuck. If you simulate it for nanosecond, you will be stuck here. How do you come out of it? You pour water. That is the bias, meaning that you are helping the system to go from one end to the other end by pouring water. You think of this guy. Initially it was stuck, you pour water, or it is raining, then now it becomes flat, and you know how much water you have poured inside. You flip it, you get the free energy surface. You know about potential energy surface, right? All of you are aware of potential energy surface. Then you add your T delta S. You think of Gibbs-Helmholtz equation. Delta G equal to delta H minus T delta S. T delta S if you add, then this is your free energy surface going from one basin to the another basin. You think of exothermic reaction, endothermic reaction. Then you have in the free energy setup endogenic and exogenic, this kind of reactions, right? You go from one basin to other basin, and delta G negative or positive, depending on that, you know the feasibility of the reaction. And these are some advanced stuff that how we construct the bias. So that means how we construct the water, how much we are pouring, and how we extract the free energy from that. Okay. Now this is kind of a summary. DFT, we are here with few atoms we can simulate for time scale something like picosecond. The length scale is also nanosecond, and time scale is also something called picosecond or nanometer and picosecond. Classical MD, you see here, typically you take nanoparticles or nanoscale, and time scale is also something like nanosecond to microsecond or even millisecond you can go. Then you need to take the help of coarse grain models, by which you can take bigger system and you can run it for longer time scale. And machine learning based simulation combines these two. So today I am going to talk about some applications, which is how a nanocluster or nanoparticle forms. Okay, all of you are aware of nanoparticles, right? But then we are curious how the nanoparticle forms. We know how you see the bubbles come out of your reactor, but how it catalyzes the process, heterogeneous catalysis. How a drug molecule which looks like this, peptide based drug, is gets delivered to the cell membrane. These are the things that I will be discussing in next 15 minutes or so. Again, the summary: normal molecular dynamics, enhanced sampling, you go back and forth. You can study the self-assembly process, apply quantum mechanics, then you can study catalysis happening on nano surface, and then you can use a neural network to do a lot of interesting stuff. Now coming to metal nanocluster. Okay, metal nanoclusters are, again, the meaning is you deal with metals. Nanoclusters means the size should be less than 100 nanometer. That's the definition typically. But the one that I will be talking about is what Professor Pradeep is expertised. I mean, he's a pioneer of this nanocluster field. is a very tiny one, 2 nanometer, 3 nanometer. Okay, nanoclusters, and sometimes to stabilize this small nanocluster, you need ligand molecules. So, these are bulk structure, the one that we see, right? The materials that we are seeing here, these are bulk materials. You think of nanoparticles, so typically in this range, you go, make it smaller, and then you get nanocluster, and then you go to atom. So this is a kind of spectrum. Depending on that, either you deal with bands, or you deal with molecular orbitals, or you deal with atomic orbitals. So again, this is a kind of textbook stuff, going from atomic orbitals, molecular, band, and then you talk about electronic state structures. So this is one type of nanocluster which is monolayer protected, meaning that you have the core, which is again certain number of metal atoms surrounded by, or rather protected by, ligand. So typically these are organic molecular ligand which are coated on top of these metals through thiol bonds. These are atomically precise, they have well defined structure, they are ligand protected and well defined molecular mass. I mean, these are important for chemistry as well as how we characterize it. Their applications: drug delivery, catalysis, self assembled material for certain device and sensors. Now coming to how a nanocluster is born. So birth of a nanocluster, again using all the tools that I discussed, machine learning based molecular dynamic simulations. Okay, so this is our final nanocluster that we see how it evolves in molecular dynamic simulation. Let's see the movie first. Yeah, this is fine. This is my student, PhD student, who is about to graduate. He did the work. Now this movie tells you, so initially we dispersed all the things, all the precursors, that means your test tube or beaker, you put everything there, and then you see how the particles, so these are, the metal is colored with yellow color, this is silver I think, and this is the ligand, and how they form the dimer. You see instantaneously they are forming a dimer, and there is a time. Now you see the time step, that same time step I was talking about, Newtonian. I am walking like this way, same time step, and what we are monitoring is some property, how they are evolving. So there is some property, and you see after some time, after this 300 picosecond, we saw one star forming here. Okay, this is what we observe in molecular dynamics. So again, this is extremely difficult to understand by doing experiment, which can be understood by running molecular dynamics simulation. Okay. These are again I discussed. So these are the tools that we have used to do the stuff. Now, what is the mechanism? We saw the movie, right? Now we need to understand what happens. The movie is just to observe it for visual satisfaction, but then we have to do some kind of post-mortem of this movie. What happens? There is a monomer which is silver connected to a ligand. It quickly goes to dimer, and then these dimers, three of them come together to form this molecular cluster. That was the expected mechanism, and that's why the intuition is. Initially we thought that this is how things would happen. Okay, and if your intuition is right, then you get excited once your intuition comes out to be right. So imagination, intuition, imagination, and then eventually execution. These are three extremely important components in research. Other option is like dekhte hai, we mix it and see what happens. Okay, that is another thing, but you get more excited if you imagine the process and that comes out to be real. Okay, then what we observe that indeed in the simulation we saw this process happening from the monomer to dimer to this octahedral complex, and this is a downhill process. So this is a typical free energy surface. You have seen potential energy surface, but with T and delta is because this is real time, real temperature and entropy, everything is there. It's more realistic. If you go from D, which is your monomer, and it's a downhill, that means this is deeper. It's a downhill process. When you leave the monomers out there, quickly forms the dimer because it's a downhill. It's a delta G negative. Now you leave the dimers here, and then it quickly goes to the octahedral, the final star process. Again, what we observe, these methanol molecules, initially these guys have to leave to allow the solid molecules come together to form the octahedral. So that's something very interesting. It's a de-weighting. Okay, so meaning when the solutes come together, the solvent molecules leave, should leave. That's the process, and that's why it is L-shaped. Solvent removal followed by solute attachment. That's the mechanism. Okay, so this is kind of a summary of the stuff. Now, once the nanocluster forms, how do they behave in solution? Okay, so how do they behave in condensed phase, solution, or in the crystalline phase? So there comes to to study their self assembly. Okay, now to understand self assembly, the first process we talk about self assembly, right? The first process of self assembly is a dimerization. It's a dimerization is the first process. So dimerization, and then the next would come, and the fourth one, fifth one, like that. Okay. So we wanted to study how the dimer forms. We put these two nanocluster nearby, and we saw the ligand molecules talk to each other. The way you imagine yourself, right? When you talk to somebody, we spread our handshake karte hai na? It's the same thing. So the way they interact is through hydrogen bond as well as pi bond. And when these guys interact, the other guys, other ligands, they move backside. It's called snorkeling effect. You know this one? Water, when you go for this thing. So this is called a snorkeling effect. So that means essentially when you spread this hand, the other hand goes back. So that's the idea. And now we applied enhanced sampling method because it's a barrier, it has a barrier to cross, and we studied enhanced sampling and see how these guys come together, form the dimer, and goes back and forth from monomeric to dimeric states many times, and from there we calculate the free energy surface, and it turns out that the dimer is stable by almost 50 to 60 kilojoule per mole. Okay, so that means what is the idea? So from here you know that if you take this nanocluster in the lab with some saturation, leave it, it will form some self-assembled structure because your delta G is feasible, it's negative. We did a bit of pH dependence. It's not really, it's kind of mimicking pH because we tweaked the ligand charges because this is carboxylic acid. So we can have carboxylic acid or carboxylate depending on the pH, and we found out that around 25% deprotonated state is the best one to form the self-assembled state. So these are again computer prediction. No, it will not stop. The dimer was just to understand the initial step. Now we leave everything in the solution, in the soup. Now you see these guys are forming, so these are sodium cations, they accelerate the process. Now they form this kind of structure. You see this kind of linear, it kind of form bands, it kind of shows some pattern. Okay, but this is again initial studies. Then we thought, what if, how do the ligands molecule talk to each other in the crystalline phase? So we set it up one FCC crystal of this particular nanocluster, and see it's not so static. We assume that in crystal everything is static, but these guys, once in a while they switch and interact with each other. It's not plastic, but it's not like so static crystal. They can still have some reorientation in the crystalline phase. So this was our first attempt to understand nanocluster few years back. Then once you know that things you are under control of doing things, you imagine, what if I have a nanocluster, one side of it, half of it positively charged and half negatively charged. That was a thought, imagination. This nanocluster has not been synthesized yet. It's called Janus nanocluster. That means half side is positive, half side is negative. Assume this red color is negative and this is positive. Now you can think of a spin or a dipole moment, or say dipole. So without doing anything, you know how the dipole would be aligned, right? They can be aligned like plus minus plus minus plus minus, like that, linear. It can have a triangular one also, but with some level of frustration. You can have benzene like this kind of thing. This you do on the paper before doing anything. We simulated it and we found out that these guys indeed form this kind of pattern. Okay, I'm not going to discuss in details, but you can look at our papers and you see this kind of automatically, you leave everything in the solution, it forms this kind of structure very quickly. And then we recently studied how if you incorporate some level of chirality in the system. Okay, so this nanocluster is not chiral, but the way they aggregate, it forms kind of helical structure. And we have studied how each particle, one particle comes and sits here, and then helical, it grows like this. So this is again imagination, and eventually it ended up we ended up getting something circular as well as this helical pattern, and that matches with, to some extent, again the length scale are different, with a paper recently published. We followed this paper and modeled it. So this is, you see, like here, you coil them and it formed this kind of structure. Okay. Then we thought nanoclusters are promising in catalysis. Okay. It almost mimics like enzyme. How? If I show you this structure, then probably you realize, where is it? Let me see if, look at this. If I show the nanocluster in this representation, okay, with volume, surface and those kind of thing, then the ligand molecules which are shown in blue color, there are some kind of pockets. This reminds you of this Pac-Man type of model that we study in protein, right? So there is a protein, the ligand goes and reaction takes place and product forms. This is also almost like a enzyme mimic. We thought that, okay, let's model something here. Let's model one chemical reaction mimicking, let's say this is almost like enzyme mimic. So we took this again, this is a published paper in SES catalysis, and we wanted to study how the cyclization takes place inside this nanocluster using computer. So before doing that, we did something in two different solvents, and that we found out that in chloroform these pockets form very quickly. That means this part is exposed where the catalysis may take place. Okay, but here it is random, so it's not allowing, it will probably not allow the substrate to sit in, but these guys can allow the substrate to sit here as well as here, then reaction would take place. So we went ahead with this chloroform setup, chloroform solvent, and what we modeled is something like this. This is a substrate which is acyclic, and you'll see this guy oxygen took one proton from here, one hydrogen from here, another hydrogen it would take from here, and from H2O2. So oxygen goes from oxygen to H2O2, and this ring gets cyclized. If you follow this, oxygen first it abstract a proton from here, next one from here, and then this guy O negative goes and binds and it forms a cyclized product. Okay, so H2O2, and now you see it's cyclized. This is assisted by a ligand here. That's the beauty of it. This reaction can happen in solution, yeah, but with a high barrier. Now this guy kind of stabilizes the intermediate by some hydrogen bond in between. That's how it is catalyzing the chemical reaction. That's the beauty of doing molecular dynamics and seeing things at the molecular level. And then what is the next thing? Because this is already experimentally done. So what have we got from the here? We can change this ligand. Now we can design. So change the ligand or ligand engineering, and we can design our reaction. And that would probably allow us to design some nanocluster for some target chemical reactions. Okay. Now if you want to understand what is the energetics of it, look, the transition state is here. This is tiny barrier, so that means it's a catalysis process. Okay, so you could find out delta G and all the transition states and everything. So I'll quickly go in 2 minutes and then I'll stop. But then another type of catalysis, which is heterogeneous catalysis on surface. Here also we took a magnesium oxide surface and I put a tiny nanocluster. It's a gold 8, but we created one defect intentionally to make things more complicated. So this defect, again you have studied in solid state, is called F-center defect, meaning that you remove one oxygen, so you get some electron localized here, and that does some magic. Okay. So and the reaction that we are studying is CO oxidation. So carbon monoxide, you add oxygen, you get carbon dioxide. This is an important process. So this is the system that we study, and this is the reaction that we are interested in. Now, when we created the defect, there was one purpose. The purpose was that we put the nanocluster on magnesium oxide, and with the defect, how does this guy behave near the defect? It turns out that this gold atom gets anchored, attached to that epicenter defect, and it remains there. Okay, look, this is almost anchoring, holding this nanocluster on the defect. Another interesting thing is, if you look at the geometry of this nanocluster, it's a two-dimensional seat-like structure. So previously, a lot of people have studied this system 30 years back. Okay, some pioneers in the field, they have studied this system almost 20-30 years back, and they claimed that this three-dimensional structure is dominant one based on some Gaussian, some gas phase calculations. But then this is not the case. When you apply real temperature, real pressure, and entropy, so this simulation is realistic thanks to machine learning. Machine learning allowed us to see things happening in real time, real temperature, and that revealed that it's not the three-dimensional one, rather two-dimensional one are dominant. And again, if you look at the free energy minimum, you see that these structures are here. That's a new thing, okay, with the AI changing the whole spectrum of molecular dynamics. Now that this is anchored properly, this is your active state of the nanocluster. It is localized. What it can do is it can bind carbon monoxide through this peripheral gold, and then the reaction takes place. CO is approaching slowly to this guy, and you will see at some point that this guy, CO gets converted to carbon dioxide, and this oxygen gets anchored to magnesium. In the next cycle, this gets utilized for another carbon dioxide. Okay, so there are certain mechanism. MPCs again we utilize for drug carrier. So very quickly, this metal nanocluster, this is a peptide based drug that can kill cancer cell. This is almost like a needle. You think of a cancer cell whose membrane is very weak, you bring one needle and burst it, like busting a balloon with a needle. That's the idea. What we studied is, so this is an experimental paper which shows, kind of speculated this might happen. What we observed in our case, that this peptide gets anchored to the nanocluster through this positively charged amino acids and very stable, and that allows this nanocluster to carry this peptide to the cell membrane. We have found out this energy barrier is not a trivial thing, it's a 50-60 kilojoule. That means this guy remains here and it carries like this to the cell membrane. And what we did here is, now you remember the coarse grain model, so we reduced the number of particles that allowed us to study this process. Look, the nanocluster is coated with the peptides shown in this different color, how it is delivering this peptide to the cancerous cell membrane. This again molecular dynamic simulation, by which eventually it gets here, this kind of peptide anchored here, and it disrupts the whole membrane. That means it's puncturing, it's creating a hole, and that way all the intracellular stuff would come out and eventually the cell will die. That was the process. So in summary, using computer simulation, starting from looking at the water in water bottle, we could go all the way to modeling drug delivery or catalysis, self assembly, how the nanocluster forms, catalysis, and drug carrier. These are all kind of things can be done using those set of equations and algorithms. That's the beauty of molecular dynamics. And once in a while, or very often, we collaborate with experimentalists, including Professor Pradeep. Whenever things are like we don't understand, like whether this is real or something can be utilized, we predict something and then talk to experimentalists, see whether this can be done. We have designed that can be synthesized or not, or the other way around. They have done something, they're not able to see things at the atomic level, then we do molecular dynamic simulations. Is complementary on both sides, and that way we could do things, some exciting science with this. So by the way, this is our website. If you're interested, team, all the codes and kind of things, tutorials would be populated. You'll find something here. It's not there, but if you go here, they'll find the codes. And this is my group at IIT Delhi. So these are the guys who does all the magical stuff. Okay, and the funding agencies for support. And thank you all for your attention. Sorry, I took little extra.
Nice presentation.
Thank you.
Actually, I have a question. in Gaussian simulation, we have run for the HOMO and LUMO and MESP also. The HOMO and LUMO, there is a two color region, red and blue. The blue color region represents as a positively charged and red is a negatively charged. And come to the MESP, like molecular electrostatic potential analysis, it is totally different. Like positively charged region is represented as a red region and negatively charged region as a blue region. Why?
Oh, okay. So this is kind of a choice. Huh? So there are two things. Homolumo is standard. Okay. When you talk about molecular electrostatic potential, you can choose the range. I mean, you can choose whether you describe your positive by blue or red. That is your choice. It's not like it's a convention. No. There's another convention that crystallographer use when they talk about density difference or some electron density difference or charge difference. Then you have to check whether they are plotting the difference as red or blue or they are plotting some absolute number. So this is kind of a convention people use interchangeably, but there is no science in this, but it's just a choice.
It depends upon the molecules.
No, it doesn't depend on the molecule. The way you choose the description. Okay. Not the basis set. So you want to show the positive from the molecular electrostatic potential. You want to show the positively charged in blue. You can do that. Somebody else can do it in red color. Somebody else can do it. But the meaning should not change. You need to describe that I have chosen like this. There are some conventions where people talk about charge difference. So if the charge has increased, you can use a blue color for the increase in charge. For the decrease in charge, you can use a red color. These are like your choice, but it doesn't change the physical properties of the molecule. Yeah.
Thank you, sir.
Yeah.
It was a very good lecture. I just wanted to ask for a small doubt. Like when you told about the cancer treatment,
Yeah.
it acts as a needle. So I was just curious that there are two kinds of cell death, right? Apoptosis and necrosis.
Yes.
So if we do it like a needle, then it might be more possible that necrosis will happen. That is not desirable. So did you check that?
Yeah. So in this case, the experimentalist, they actually told that the way it is done. So typically, when you go to the membrane, right, you take a nanocluster, it can diffuse directly through diffusion, or you can form this kind of a this bubble. What do you call it? Endocytosis, right? Endocytosis. So when you bring this metal nanocluster to the membrane, either it can form this or it can follow the cytosis pathway. That means it forms a small bubble and then it brings the whole thing inside, or it can directly diffuse as a direct diffusion. In this case, they claimed or they showed that the peptide goes to the membrane, two or three of them, and then they create a pore. So if you think of aquaporins, so this peptide should align like this vertically and then move the lipids little aside, and that creates a kind of a puncture in a tire. And when you create the puncture, then whole intracellular entity would come out. That's how the cell is died.
But it whole it will come out and it will poison the environment.
It can, but then cell debris eventually gets out, right? So let's say on the next cell would eventually, these guys, if they go inside the cell, then there is something, what is it? Huh? Lysosomal activity, exactly. Lysosome would eventually cluster them and then.
Actually, that happens in apoptosis, right?
Okay.
Like programmed the way it gets disposed. But this is kind of accidental cell death that we are creating here.
It could be because I don't know the exact pathway of this post cellular death situation, but looks like that once everything gets out, water and this kind of cytoplasm, this thing would eventually come out. But whatever the other rest of the things, if they penetrate, yeah, yes. Or sometimes it would not even allow the mutations to happen so rapidly because these guys.
I think the effect it's coming due to the APR effect.
Yeah.
Whenever the cancer progress, the APR effect for enhanced permeability retention.
Yeah.
The acidic condition, the site of cancer region was increased,
Yes.
which increased the vasculations. Means
Yes.
the size of membrane, if it is up to the 100 nanometer, it goes to up to 800 or 1 micrometer.
Yes.
So due to the vascularization nature, these molecules can penetrate
Yeah.
inside the
Yeah.
membrane, can target the cancer regions
Yes.
without doing any
Yeah.
necrosis.
Yeah, in layman language, the membranes are very weak and they're kind of swelled, so it can penetrate quite quickly. Other than your normal cells, because they should not harm your normal cells. Yes, please.
What is the time scale of the simulations that you perform? Is it same for all the simulations or it depends on the system that you simulate?
Depends on the system and process.
What is the maximum simulation window that you want to like the hardest?
Let's say, because these are all enhanced sampling. So in enhanced sampling, if you run 1 microsecond, that would scale to few seconds. Okay, so that scaling we didn't do, but the longest one is typically something 20 microseconds. Yeah, it's not one, multiple of them. Yeah.
So from an experimentalist perspective,
Yeah.
to corroborate this theoretical data, if I were to set up a spectroscopic experiment, a time-resolved spectroscopic experiment, can we look at this picture from that perspective and like bridge the theoretical and the experimental? Can we do that? Can we set up an experiment like that and then try to like understand your data?
Yeah, in fact, I'm talking to some ultra fast people or SFG, those kind of, to see what happens at the interface, because the interface catalysis that when you do, probably some frequency generation that kind of would help. And then we are doing something called nanocluster under confinement, which Sukhendu we are doing. So you take a MOF and you put the nanocluster inside and the reaction takes place. So there we are thinking of utilizing ultra fast to see whether we can capture the signal during the some conformation inside.
Yeah, what you are doing is actually very amazing and very interesting. I just to get the actual, just to like corroborate, bridging is very important.
Yeah, in fact,
experimental and theoretical work together.
In fact, this nanocluster nucleation, it would be amazing if we can get a liquid cell in situ TM, right, or something like that. But we don't have the setup here.
Work in 2D spectroscopy and they do MD and 2D together to corroborate. So if you could do something like that, it would be very interesting.
Yeah, I can tell you that some guys are doing abroad, but in India, if we start, that would be next level. But I'm really hopeful with this, Professor Pradeep. Things would change at some point.
Okay.
Yeah.
My question is, how do nanocluster activate small molecule like H2O2 CO2?
So there are two ways to do that. One way is, let's say, thermal activation. It's mostly deal with charges. Okay, or even if you take it at room temperature, assume that the reaction is happening room temperature, the charge redistribution, the one that I showed you, takes place at room temperature. Charge redistribution that takes place on the nanocluster because of that defect, epicenter defect, actually activates your oxygen-oxygen bond in the carbon monoxide oxidation. Epicenter defect. So the charges that get localized at the epicenter changes the whole charge distribution on the nanocluster. That charge transfer also takes place to oxygen. That's a huge charge transfer. I didn't show it here, but I have slides which shows that. But other side of it is like photosensitizer. If you shine light on top of it, then it activates oxygen, and then it does the reaction in the cyclization process. Yeah, it works like as a photosensitizer.
So what is quite clear is that nanoclusters have done a lot of good for you.
Yes, indeed.
That's very happy to see.
Thank you.
And many good things have come out, and we'll now that everything has been set up,
Yes.
more collaborations and more activities will happen. It's great to see how this is evolving.
Yes.
But this is also, I think, this talk for the students. I think this is in a way build the right kind of pedagogical framework for something to be looked at.
Yes.
Obviously, your teaching excellence has come out very clearly
Thank you.
from this. I was just before this, we were talking about opportunities. So maybe you've also gotten a message as to how to present yourself, especially when you look for opportunities, jobs elsewhere. There is something to collect from all of these as well. Hopefully, you've added that component as well
Thank you.
to the discussion. I have nothing specific to talk about. I was looking forward to some data on the cryo-EM,
Okay.
which you would probably show some movie I was thinking. In fact, it is there really.
I
As you
have
just
slides.
now
Yeah.
talked about.
Yeah.
That is how these clusters are penetrating.
Yes.
So these are things that today cryosectioning can reveal.
Yes.
The very same molecule which is attached onto a cluster when it is delivered, sections of these cells can be taken.
Yes.
No one has so far done that with clusters. Well, this is very much possible.
Yes. Yeah.
Thank you. I have nothing more to.
Yeah. Thank you. Thanks a lot.
Your model can we rationalize the synthesis of nanocluster? If I want a, let's say, AU22I1, and you can predict that which means the composition of metal and ligand.
Yeah, so in fact, I can tell you now that we are developing a method by which we can do it without simulation. So this is based on something called generative AI.
So he's saying that you mix all these things together, it makes only 22, not 25.
Yeah. So it can. So what we are doing is we are taking metal combinations, ligand, or all sorts of experimental conditions to predict the final product. So just give us few months, the method would come out. Yeah, we are working on it. Yeah. I mean.
Thank you, sir, for showing that how modeling turns complexity into clarity. Thank you.
Sure. Thank you. Thanks a lot. Thanks everyone.
Wow, you are putting everything into